Yeah, the group of bacteria that are different from the others you have covered so far, mostly in their shape, and also one cannot do a gram stain on them. So they have their own unique features, and even the illnesses they cause, these are different from the commonly encountered hygienic bacterial infections. So we are going to describe them in general and then narrow down to the genera that involve human beings. We are going to describe those briefly, maybe just a little, like we are going to talk about their clinical significance, the way they are transmitted, and the way they are seen in the laboratory. So these are bacteria which are grouped under the title Spirochaetes, and that actually reflects their shape. And their shape is such that they are spiral, and that word is derived from a Greek word for hair. So it also implies that they are very narrow. So they are narrow and spiral organisms. So there are many spirochetes, but we're just going to mention the significant ones, which are related to human infections. But generally, spirochetes are bacteria which are microscopically, they are narrow and elongated. They are spiral, and they are tight coils, making that spiral. And if one cuts across it, they have a cylindrical shape and they have tapered ends. These organisms contain in them some structures which are made of the protein that the flagella that you're aware of are made of, that is flagellum. But they are fibrils within that long organism. So they are referred to as internal flagella. And they are many, they are attached at both ends, and the number varies according to the species. They are also known as axial fibrils, and their function is more or less the same as that of flagella. In other words, they are organelles, or they enable the organism to move actively. So they are organelles for motility, or part of the organism that makes it move. And it can move in a way that it makes some progress, but it can also move on its own axis, where it just moves without making any uh, progress. That is rotational movement. So structurally, also, they possess several similarities with gram-negative bacteria, and they also have a few differences. So they are not typical gram-negative bacteria, but they are closer to the gram-negative organisms than gram-positive. So among the differences, they don't have that important characteristic of gram-negative organisms, that is possession of lipopolysaccharide in the cell wall. So that one is absent. And then, unlike the gram-negative organisms that you're familiar with, that are easily stained using the gram stain, these particular ones are not easily stained. So with that general introduction, we can look at some spirochetes associated with human infection. And these ones fall in their three genera. One is Treponema, another one is Borrelia, and the genus Leptospira. So if we look at, we start with the genus Treponema. As a genus, it covers many organisms, and it's a mixture. There are some which are pathogenic to humans, and there are some which are non-pathogenic to humans. So among the non-pathogenic ones, species of Treponema, there are some which are encountered as saprophytes. Just a second, please. So there are some which are encountered as saprophytes. They are referred to as saprophytes, but they are different from the saprophytic organisms we have mentioned in the past, in that they are not found freely living in the environment. They are kept in laboratories. They are actually developed there for study. And one of them is a species, or all both we are going to mention. So there are two which are well known, which are actually strains of the pathogenic species of Treponema pallidum. So we are going to talk about that pathogenic strain in a short while, but as of now, there is one which is a strain of it that is not pathogenic, that is developed in the laboratory for laboratory studies. And they are maintained in the laboratory and used for studies because these are very thin organisms, very delicate, and the pathogenic one is difficult to manipulate. So most of the information that we have is derived from this particular organism. And it is also different from the pathogenic strain in that it can be grown artificially in culture media. Another group of non-pathogenic Treponema species occur as normal flora. They have their specific names, but there are few of them encountered as normal flora. Some are found in the mouth and grouped together with other spirochetes in the mouth, and are referred to as oral cavity spirochetes as part of the normal flora there. And then there are also a few in the genitourinary tract, the lower part where normal flora are found in small numbers. Some of them are spirochetes. So that's the non-pathogenic organisms or species. If you look at the pathogenic species of Treponema, the main ones are Treponema pallidum, Treponema patinui, and Treponema caretium. So these have been the naming of those species, but some people have come up with a classification. So depending on what you are reading, you'll find that some of these organisms have been declassified as um, subspecies of Treponema pallidum. In that what was formerly called Treponema pallidum is now Treponema pallidum, subspecies pallidum, which we just call Treponema pallidum. Then there's Treponema pallidum, subspecies patinui, and Treponema pallidum, subspecies caretium. So you can either look at them as subspecies of Treponema pallidum, or you can just look at them as they were in the past, like as Treponema pallidum, Treponema patinui, and Treponema caretium. So either way, it's acceptable. All these three species or subspecies are morphologically similar, so one cannot distinguish them by looking at them under the microscope. And they also possess some antigens that are similar, because when they form antibodies, an antibody formed by Treponema patinui can react with Treponema pallidum, and vice versa, and all the way. They cross reactions in serological tests. That means they contain similar antigens. So to differentiate them, sometimes one has to go by geographic location. There are some which are known to occur more in certain parts, geographical regions than others. And then there are the clinical manifestations, although they share a lot of clinical manifestations, there are few differences. So those who handle those patients, they know some minute details of the clinical manifestations that are caused by one and not the other. And thirdly, where they can analyze the genetic components, then that too is useful in differentiating those species. So there's also an organism that is documented now and then. It is Treponema denticola. It is one of the normal flora spirochetes in the mouth, and it has been encountered occasionally as one of the causative agents of opportunistic infections in the mouth. So it's not a very prominent pathogen or pathogenic species, but some people have encountered it and have documented the importance of this particular Treponema species. So if we look at the Treponema pallidum, the one that was well known as Treponema pallidum, it is a narrow, elongated spiral organism with tight, regular spirals and tapered ends. Again, that description would make sense to people who see all of them at the same time. So they can gauge the regularity of the spirals and the tightness. So with that, they can see the differences and they can identify the species. Otherwise, it's too narrow to be seen by gram staining and commonly used light microscope. So if it is present in a specimen, maybe some fluid, and one makes a smear and makes a gram stain, one still would not see this organism, although it's present. For practical purposes, it can be demonstrated by one, a special type of a microscope referred to as dark field microscope. This particular microscope has a special condenser that limits or cuts off the light that would enter through the objective, through the condenser. So whatever is seen through the objective is the organism reflecting the light from the surrounding. So it gives you a dark background because of the condenser, and we are able to see the organism making its way because it reflects some light and shines. And it is recognized by its shape and fertility. Secondly, it can also be demonstrated using fluorescent antibody staining and fluorescence microscope. So that again is not available commonly, but where it is available, one can comfortably have mentioned that with regard to other organisms, because it's based on antigen-antibody reaction. That is, one has an antibody that is attached to some fluorescent material and reacts with an antigen on the body of that particular spirochete, and it remains there. And so one sees the fluorescence, and using that one can see the Treponema pallidum, subspecies pallidum. Then thirdly, staining in tissues by special techniques and microscopy. This particular one is not done in microbiology. It would be done in a laboratory where tissues are processed and stained. So that is a totally different area, but it demonstrates the presence of the organism in those tissues using those special staining methods. And fourthly, there's electron microscope. That particular one again is more rare. It's found in maybe in research institutions and some special laboratories, but where it is available, it can be used to view Treponema pallidum, subspecies pallidum. Growth of the organism in the laboratory. It's one organism that does not grow artificially. So attempts to grow it or to culture it in artificial media remain unsuccessful. But there are some situations where some workers uh, in the past they've isolated an organism, or they can get it from subdue it from a patient and they would like to propagate it. There are some suitable laboratory animals that can be used or have been used in the past. They use some specific parts of that animal and the organism is injected or inoculated with those parts and it multiplies there and they are able to study it more. But the disease that this organism causes in the humans is a very severe type illness and it does not manifest or it rarely manifests in those animals. I guess it's just for a while, but it can't go on for too long if the interest is mainly to get the organisms. So it is done and it's recorded that the animals develop the type of illness that is seen in humans. Then the significant physical properties are usually these physical properties that will help one understand possibilities of transmission. So for this organism, it is small, it is very delicate and very narrow, so it is highly sensitive to physical changes in the immediate environment. It doesn't last long. If it's deposited somewhere outside the body, it dries up very quickly. So it's rendered non-viable by exposure to dry conditions. It is susceptible to heat, like other bacteria, and cold temperatures also would affect its viability. It can remain viable at the refrigeration temperature, that is 0 to 4 degrees for up to 2 days. But after 2 days, then it becomes non-viable. And this particular information is significant for fluids, maybe blood, which is limited. If it is kept in the refrigeration temperature, if it contains Treponema pallidum, those organisms can be viable in the first 2 days, and then after that it becomes non-viable. But even if it contains organisms which are non-viable, it still should not be used, or it's not used. But it's important information that should that blood be transfused into someone within 1 to 2 days, there's a possibility of it containing the spirochetes which are still viable. Thirdly, also with regard to physical properties, it's an organism that is susceptible to antibiotics, but there are also some chemicals, various chemicals, and these were used to treat the infection before the antibiotics were developed. So if you wish to know which chemicals these are, then you can look up. Antigenic properties and antibodies against that organism. So when it comes to its antigenic properties, it is known that they exist, but picking on them and analyzing them has proved difficult. Basically, again, because of the delicate nature of the organism, dissecting it and getting these antigens and studying them has proved to be difficult and it has not been done. But it is known that it has antigens on its surface because when a person is exposed to it, the immune system recognizes these antigenic parts and responds by forming antibodies. So the exact antigenic components are not clearly identified, but the immune system is efficiently stimulated. And what stimulates the immune system to produce antibodies are antigens. So it is efficiently stimulated and distinct antibodies are produced in response to infection. So these antibodies, they can be detected in the blood of the person who has been exposed. There are quite a number of them, but we are going to talk about two because these are relevant to us when we are doing the laboratory investigation. One antibody that is important is an antibody which reacts with a substance from outside. It is called cardiolipin. It is it reacts with cardiolipin, and this cardiolipin is a lipid compound which is extracted from deep heart muscle. So this is done scientifically, and it is packaged scientifically such that it can be used in the laboratory on a patient's serum to detect this antibody, the antibody that reacts with cardiolipin. So the antibody has many names. Among those names is Wassermann's antibody, or reagin antibody, or anti-cardiolipin antibody. So it is the human is exposed to the organism, and the body reacts and produces an antibody which can react with other things. And we are going to mention again that this antibody can also be produced by other infections. But what is important to us is that when a person is exposed to Treponema pallidum, subspecies pallidum, in one way or the other, there's an antigen somewhere that makes that person produce an antibody that reacts with cardiolipin. Secondly, there's an antibody that is formed that is more specific. It reacts, it is due to a specific antigen within the spirochete. And so the antibody that is formed is referred to as anti-treponema antibody. So the clinical significance of Treponema pallidum, subspecies pallidum, the natural infection is limited to humans. It's an organism that only infects humans naturally, and it is maintained in life by humans. And the illness that it causes is called syphilis. It is an illness that has been there for many years, has many centuries. And it is a severe illness with very severe complications. So the transmission of Treponema pallidum, subspecies pallidum, depends on the presence of it in the blood circulation or in the lesions due to the disease, some of the lesions. Main methods of transmission, one is sexual transmission. It's one of the sexually transmitted infections or sexually transmitted diseases or venereal disease. Secondly, it can be transmitted by an expectant mother. If she be infected from her blood, she can transmit the organism to the fetus or the expected baby. And the illness is referred to as congenital. And thirdly, there's a possibility of transmitting the organism via blood if it is donated by a person who has the spirochetes in the blood, and it's transfused when the spirochetes are still viable into a recipient. But this particular one now is not prominent. It has mechanisms have been put in place in such a way that the blood is examined or tested for various infections, among them the Treponema pallidum. And if should blood be infected, then it is discarded, it's not used. So the third one is now a very rare possibility, but it's good to know that it's a possibility. So if we look at the sexually transmitted infection, in a situation where a person is exposed to Treponema pallidum. So the lesion containing the spirochetes is on the skin or mucous membranes of the external genital part, whether it's a male or a female, for most infections. There might be other infections that occur elsewhere in the body, depending on the type of sex that is practiced. But for most people, it's heterosexual sex That is what we are going to try and talk about, because that is what is well known. So there's an individual with a lesion on the external genital part due to the infection, and then that person gets into contact with another person, and the organisms would enter that susceptible person through broken mucosal surface or the skin, and that would not be something that is a big wound on that susceptible person's genital part. It could be a small tear or a very shallow groove, but because of the very small organism, very narrow, it gets in somehow. So it multiplies at the site of entry, and at that site, the body of that person responds by mounting an inflammatory response. So obviously there will be some inflammatory process causing a swelling and some fluid in it, which eventually we are going to talk about in a moment. But there is an incubation period which follows that exposure before one sees the clinical manifestation. And these clinical manifestations, generally they change as the illness progresses. So they change with time. And based on that, if one is to have clear-cut divisions, which is not there, there could be an overlap. Some of them might not show up so clearly, but generally there are 4 stages. Some people give them as 3, but there's one stage that can be termed primary syphilis based on its manifestations, the secondary syphilis, the tertiary syphilis, and the late syphilis. For some people, tertiary syphilis and late syphilis, they are just referred to as later stages of syphilis. But as I said earlier, practically, these stages might not be so obvious. Some of them, like from secondary onwards, they might overlap. Some of them might not show clearly, but for discussion purposes, we need to understand what happens. So for the primary stage, that is primary syphilis, during that time, there is invasion of mucous membranes by the organism and initial dissemination through lymphatics and systemic blood circulation starts. But what is seen as clinical manifestations, this can be seen within 10 to 90 days after exposure. And what is seen is a lesion that is referred to as primary sore, which is actually an ulcer or primary ulcer. It can be called a chancre or hard chancre, just to differentiate it from the soft chancre, which is caused by Haemophilus ducreyi. The commonest site is the external genital part. We say the susceptible person develop some tears, or very small tears, or bruises, very minute abnormality on the surface of the skin and mucous membrane. So that site where it gets into is where this uh, primary lesion, primary sore or chancre develops. So the commonest site is the external genital part. It's painless with clearly defined margins, and for most people, they also develop enlarged inguinal lymph nodes related to the parts where the ulcer is. It could be on one side. But because it's painless, the person continues with his life. Most people don't look for medical attention, and they don't receive any treatment. Should any person look for treatment, they will be given a diagnosis will be made and appropriate treatment given. But even where there's no treatment, it heals spontaneously on its own by the body's own mechanisms of healing, and that happens 3 to 8 weeks in the absence of treatment. And for some people, that when that happens, even the organisms are eliminated from their body. and naturally they are healed. But for some people, the ulcer heals, but the organism remains somewhere within the body. Then secondary syphilis, this particular stage, it is characterized by a large number of the organism in the blood because they remain after the primary, after the ulcer are healed, they remain in the body and continue to multiply. So the person progresses to secondary syphilis with a large number of the organisms in the blood, and because blood goes everywhere in the body, there's widespread infection involving other tissues, including some organs, and this is through dissemination via blood. So at this stage, organisms are found in organs, in tissues, in CSF as organisms. So they are found wherever the blood is going, it deposits some of them there. So the manifestations of secondary syphilis are variable, and sometimes it can resemble other diseases. But among the manifestations, there can be a skin rash, there can be ulcers in the mouth, there can be a generalized lymph node enlargement, and there can be swellings which enlarge gradually in various parts, and, particularly in the axilla and the groin. And eventually they form wart-like lesions which resemble warts, and these particular ones are referred to as condylomata lata. As and commonly, one might have some inflammatory processes also involving other tissues like the bone, the joints, and the eye. But whatever specimen, whatever is examined, or whatever develops in secondary syphilis, it has numerous Treponema pallidum subspecies pallidum. So from there again, in the absence of treatment, the person might still recover naturally due to the body uh, healing processes or immunological mechanisms. There are some that develop, the symptoms disappear, they become asymptomatic, and they develop a symptomatic state referred to as latent syphilis. But the illness is not totally eradicated from them. Then there are some that progress to the next stage, which is tertiary. And in that tertiary stage, it comes 3 to 10 years after the primary illness, and the organisms are no longer detectable in the lesions. There might be there a few of them, but they are rarely detected. And most of the lesions now at this stage are due to chronic inflammatory processes. And they manifest as chronic inflammatory swellings referred to as gammas. And some people call them syphilitic gammas, differentiate them from other chronic inflammatory swellings, and they can occur on the skin. They can involve the mucous membranes, and they can involve the bones or other organs. And then the ones on the skin, from time to time, they can get eroded. And when they get eroded, the person develops an ulcer at that site. Then the late stage disease, clinical manifestations occur much later, 10 to 20 years after the primary disease, and the manifestations target two major systems. Two major systems. One, the cardiovascular system, and here also it's more of a disease of the large blood vessels. So mainly as inflammation and other abnormalities of the blood vessels. It can involve the aorta most of the time. It can involve that large blood vessel called the aorta. It can weaken some parts of the wall, or it can affect the aortic valve, and the person develops aortic incompetence. Then the second system is the central nervous system. And the central nervous system disease due to Treponema pallidum, subspecies pallidum, is referred to as neurosyphilis. So this is not single clinical entity or disease entity. It just implies that the person has central nervous system disease due to syphilis. The manifestations are varied and they depend on the function of the part of the central nervous system that is affected. So some patients rather, the illness may be a change or can occur as a change in the way the person walks. So it can affect the gait, it can affect the joints, they become deformed, it can affect the optic nerve function, it can interfere with the mental capacity. The person might have some mental abnormality, and it can affect the meninges and blood vessels and give rise to more complications. So clinically, you'll get to know what the terminologies are for the gait that it develops, the abnormal joints, and the optic nerve function. But for now, it's just good to know that it can be reflected. That its syphilis can affect the gait later in life. We mentioned a situation where the person carries the organism but has no symptoms after the secondary stage. It is a dormant disease, it is latent syphilis without symptoms, and it is detectable by serological tests. So it can be an incidental finding, or for one reason or the other, the clinician might request for it, but if one looks at the clinical manifestations, they're not there. The symptoms are not there. But it's important when it is detected that it is treated, because it's capable of developing into cardiovascular disease or neurosyphilis in the absence of treatment. Then we mentioned congenital syphilis resulting from mother to child transmission. It occurs transplacentally, and it can occur anytime throughout the pregnancy and even at the time of delivery. And what the baby or the fetus develops is septicemia due to Treponema pallidum. You can call it spirochaetes or septicemia. And the fetus being small, there is a widespread dissemination, and there are manifestations which are varied. It depends on how old the pregnancy is. It can cause a miscarriage. It can be a cause of death of the fetus. It can be a cause of death of a baby that is born dead. And it can also be a cause of congenital abnormalities. So for laboratory investigation, we mentioned those symptoms or signs or manifestations because they are kind of related to the specimen one would want to examine when it comes to laboratory investigation. So the methods that are used can be separated into three categories. One, there are some methods that detect the presence of the organism in a specimen. Then secondly, there are some tests which detect the antibodies we had mentioned. And thirdly, there are genetic material detection methods. Those are the molecular methods of diagnosis. They are applicable here as well. So if we look at the first one, detection of the organism, we said it's an organism that cannot be seen by doing a ground stain and microscopy. And we also talked about clinical manifestations, and we know that there are some stages where the organism is no longer reachable. One cannot get it in a specimen that easily. So it is kind of restricted detection of the organism. That is demonstration of Treponema pallidum in specimen. And it can only be achieved one in fluid or scrapings freshly collected from primary chancre or lesions of secondary syphilis. So it is the primary syphilis and the secondary syphilis that where one get Treponema pallidum, subspecies pallidum, in specimens that can be demonstrated if possible. So the specimen can be a fluid or scrapings, and it is examined under dark field microscope, and it's recognized as spiral cells with typical motility. And if we are talking about secondary syphilis having numerous organisms, it still might not be that easy to see when one is looking for it, even from the specimens. And it's done immediately because it's recognized when it is still viable, when it is motile. So it is not that easy to see them, although we are saying there are many. So sometimes the test is performed repeatedly, still one might not see the organism. Despite that, one cannot rule out the infection, that it's not there if one does not see the organism. So failure to see them in fluid or scraping by dark field microscopy does not rule out that infection. Secondly, the immunofluorescence staining and examination by fluorescence microscope. That is limited in availability, but where it is available, it can be performed. And then thirdly, staining infected tissues by special techniques, this can be done in laboratories where tissues are processed, stained and processed. So that is one category of the tests, demonstrating the presence of the organism. And you only want to do it where there is a chance of actually demonstrating it. And that chance is higher in the primary and secondary stage. Then the antibody detection, it is also referred to as serological test for syphilis or syphilis serology. They do not distinguish syphilis from infections by other Treponema species or subspecies. As we started by saying, they cross-react in serological tests. Their antigens and antibodies react with each other. It doesn't matter which organism has produced what. So the serological tests are applied on serum. They are still useful in the investigation of syphilis, despite all that. So the specimen that is used is serum in all the patients suspected of being infected by syphilis. And then cerebrospinal fluid is examined where neurosyphilis is suspected. So if one has a patient where neurosyphilis is suspected, then one would still examine the serum and also examine the CSF. Where it is just syphilis, which is not necessarily neurosyphilis, the serum is enough. So the serological tests, if we look at them, they can also be separated into two categories, relying on the antibodies we talked about earlier. So the first lot are non-treponemal tests, and these detect the anti-cardiolipin antibody, the one we mentioned first, the one we said has many names. The tests that are used to detect it are referred to as non-treponemal tests, and most sera of infected patients become positive approximately 10 to 14 days after appearance of the chancre. And there are many tests developed by very many different companies. They are administered in different ways, but the antibody they are detecting is the same. It's the anti-cardiolipin antibody. Among those many tests, we have one called venereal disease research laboratory test, abbreviated as VDRA. There's also CAN test, and there's also another test called rapid plasma reagent, abbreviated as RPR. Some people add their CAD test, rapid plasma reagent CAD test, but it's RVR, PRPR. Sorry. So there are many tests you'll encounter. Depending on the company, they'll call it something. The way it is administered might be different but what is important here is that it's in this category that detects anti-cardiolipin antibody, and it belongs to this group, non-treponemal tests. So all of them, all sera of patients with secondary syphilis produce positive results. Secondly, they are associated with false reactions. One can have false positive reactions. You see, this anti-cardiolipin antibody that is being detected can be produced by other infections. It can also be produced by some non-infectious conditions. And these are the reasons it's being detected and not syphilis, then it is a false positive reaction. Then there can also be false negative results. And we said they become positive some days after the appearance of the ulcer. So if it is done early, before those days are over, it will be negative, although the patient has syphilis. So that would be a false negative result. And they can also be negative in a patient with late stages of syphilis. So these non-treponemal tests, they are applied mostly as screening tests in the laboratory investigation. Whenever, first of all, the clinician suspects syphilis or has a reason for ordering for them, but they have that role, especially where there are many specimens to be done. They serve as a screening test to limit the number of the tests that the sera that will go on to the next stage. So for these tests, positive sera are tested by confirmatory tests. Then again, for important information about them is that the sera of patients testing positive, we say they are subjected to a second group of tests, the confirmatory test, and even for those, if they turn out to be positive, together with the clinical information, the patient is confirmed to be having syphilis. So the patient is put on treatment, and when the treatment is successful, the person is retested, and they test negative. So they revert to negative if the patient is fully treated. So in that way, they can be used to confirm the success of the treatment or to monitor the treatment. Then the second group of tests are referred to as treponemal tests. They detect the specific antibody against the specific antigen on the treponema pallidum. This is the second antibody we described, and the treponemal tests are applied as confirmatory tests on sera or CSF, which test positive in non-treponemal tests. So they are associated with fewer false positives, and the positive sera for most patients, even after treatment, they tend to remain positive. For some people have reported some that have reverted, but for most people, they find them remaining positive. And the treponemal tests, or the confirmatory tests, they include treponema pallidum hemagglutination assay. They will be shown during illustration or practical session. There's a fluorescent treponemal antibody absorption test, it's a confirmatory test, and also some ELISAs that are applied in the confirmation of the positive non-treponemal test. So those are the two serological tests or tests which are done as the serology in the diagnosis of syphilis. Then the third category of tests, which we talked about, the molecular methods, it involves genetic material detection. It could be some DNA or some RPA, depending on what it is, and the methods that are used we have covered in other areas. One of them is PCR. Antibiotic susceptibility of Treponema pallidum, subspecies pallidum. It's delicate as it is, different from other bacteria as it might be. It's still susceptible to a wide range of antimicrobial agents, among them the penicillin. The various preparations, various types or derivatives of penicillins are effective, and penicillins are the most commonly used agent for treatment. The long-acting type is particularly useful, but also the others like crystalline, they have their places in the management of Treponema pallidum. The others are tetracycline and erythromycin. So for treatment, the choice of the antibiotic, the dose, the duration, the routes of administration depend on the general status of the patient and the stage or the type of syphilis. We have talked about a disease that has many forms. So the way each form is treated is different from one another. The way one would treat the primary illness with an ulcer is not the same as the way one treats congenital syphilis or late-stage syphilis. So the details of treatment are known by the clinician, how to treat each type and which penicillin or which antibiotic. Then there are some aspects of prevention. One, it is one of the venereal diseases that we have said. So one would apply similar methods of prevention, sexually transmitted infections and STDs. So that is one advantage. These diseases are prevented in the same manner. So if one is applying the measures, then you could be protecting yourself from many other organisms or infections. They sometimes come in numbers, not one, might be quite a number. That is one advantage. So the disadvantage is the same. If one does not apply the protection method, then one might be exposed to more than one infection at a time. Secondly, the antenatal screening from diagnosis and treatment of infected expectant mothers. So if the illness can be prevented at the sexual transmission, these other ones might become less. Because even the antenatal expectant mother might have contracted the infection through sexual contact. So that prevention in the first place is very significant. That should an antenatal clinic client be suspected of being infected by this organism, then the diagnosis should be done as quickly as possible and treatment administered. That might help to treat the mother and might prevent the transmission from that mother to the baby. Then screening of donated blood that I had explained, that all blood that is donated is screened. And among the agents it's screened for is Treponema pallidum before transfusion into a recipe. So screening of donated blood is important. If the blood is detected in good time, it's not given to another person, and the transmission does not. So there are other species of Treponema pallidum or subspecies that also cause infections in humans, but these are not sexually transmitted. So they are referred to as non-venereal infections by other Treponema pallidum subspecies. So these ones are transmitted from person to person. It can be direct transmission, or it can be through contaminated items. And they can occur at any age. Some can occur even in children, and the chronic nature persists, so the person can be growing with that infection. So the organisms in this category are a subspecies of Treponema pallidum that used to be there even before the subclassification. It's called Treponema pallidum endemicum, or subspecies endemicum. And it causes an illness which is not seen in our setup. It's not reported if it is there. It's called visual or non-venereal or endemic syphilis. There are some parts of the world where it is seen often, but it's not so common here. Then there's Treponema, what was called Treponema partenui. And if you look at the subspecies classification, it will be Treponema pallidum, subspecies partenui, and the illness is yours, and Treponema caretium, or Treponema pallidum caretium, the illness is P. So you can still call it Treponema caretium or Treponema partenui, causing those, and caretium causing pinta, etc. So in general, we can't go through those illnesses one by one. They have several manifestations in common. And so much so even with the venereal syphilis we just talked about. And they have a few differences. So based on those differences, those who handle them know the differences in clinical manifestations and in the geographic areas where they are found in large numbers, they can tell. That I mentioned again at the beginning. But these lesions can be small swellings referred to as papules, and mucosal patches in the oral cavity. They can occur as nodules of the skin. These again are still small swelling but hard. They can be gammas of the skin, like what we described for the venereal syphilis. They can occur on the bones and other parts, and it can also affect the skin color. For those who are colored, the lesions will be hypopigmented, and those that are dark skin, they will lose the pigmentation. So one is the pigmentation of the skin lesions. And then there are some which are associated with extensive scar tissue formation. So you can take 5 minutes break before we go to um, the remaining genera. 5 minutes. One. cause. So we go on. Okay, so the next genus is Borrelia. Under the microscope, the structure is the same morphology. In other words, spiral organisms. These ones are described as being relatively large. In other words, they are compared under the microscope, and these particular ones look larger than the others, but they are all still microscopic. The Borrelia are not easily stainable by commonly used bacteriology laboratory stains, but there are some species that have been stained. They may stain with difficulty, and then it might require modifying or modification of some of the reagents. And when it's gram stain reagents modified, when it's applied, they, they take up the stain and they are seen as gram-negative organisms. So it's something that is done experimentally. It is not done routinely. It's through manipulating the staining reagent, and because of that, one cannot do it on a routine basis. The Borrelia are also stainable in the dry blood films. One makes a smear and dries it, and one can use Rienza stain or white stain. Again, they do not grow easily in artificial cultures, but experiments again have shown that one can actually grow them. But again, it's done experimentally, so through manipulating this and that, there has been some growth obtained, but it's something again that cannot be done routinely. So culture and sensitivity or isolation are not performed routinely, or they are not tested by clinicians for Borrelia. So we look at the species of Borrelia which infect humans. There are quite a number. One of them is known as Borrelia recurrentis. It is an organism that is transmitted to humans by body louse or body lice. The illness they cause there is called a relapsing fever. So because it's transmitted by a louse, it's called louse-borne relapsing fever or louse-borne borreliosis. Then there are other Borrelia, that's number 2. As a genus is Borrelia. They are transmitted by ticks. In this group, one is called Borrelia Treponema. Then there are others with a species name, genus name Borrelia, but the species name is the species name of the tick, the scientific name, species name of the tick that is responsible for its transmission. So those are responsible for type of uh, borreliosis or relapsing fever, referred to as tick-borne borreliosis or tick-borne relapsing fever. Then there are other Borrelia that are transmitted by ticks that I'm not going to elaborate on, and they are responsible for other diseases, which are seen mostly in temperate countries, and they are not relapsing fever. But the organism is transmitted by ticks, and it also involves animals. Where ticks are involved, there are animals involved. So we'll just restrict ourselves to the relapsing fever species. Thirdly, there are some Borrelia species which are found in the mouth. Again, they might have their specific names, but they're lumped together as oral cavity spirochetes together with other species of spirochetes. And they are significant as positive agents of ulcerative lesions in the mouth as opportunistic infection. This could occur in association with other factors like trauma. They could act like malnutrition, or they could act together with other organisms, maybe with some strict enero to produce some ulcerative lesions in the mouth. So we look at the relapsing fever, transmission and clinical significance of Borrelia species associated with the relapsing fever. We have touched on it. Borrelia recurrentis being transmitted by body louse, and these are human uh, ectoparasites. So they are from person to person. And for this to happen, there has to be some um, low levels of standards of living. So overcrowding in an environment with scarce facilities for cleanliness favors transmission of these body lice from person to person. They keep crawling from one person to another. So what happens is that the louse ingest the organism while feeding on the blood of the infected person. And then this infected person, there are many of them in that crowded area. They form the reservoir, and entering to a susceptible human is through rubbing or crushing the infected louse and its fluid onto broken skin. Then the Borrelia deutoni and the related tick-borne species that cause relapsing fever, they are transmitted by blood-sucking ticks. And they are transmitted between animals, one animal to another, and from animals to humans. And they acquire the infection through humans. Humans acquire infection either through a tick bite or by crushing the infected tick onto the skin. So again, the organisms are acquired by the ticks as they feed on the blood of infected animals. There are many, quite a number of animals, including rodents, pigs, armadillos, and even porcupines. And these infected animals, together with the infected tick, constitute the reservoir. And the infected ticks remain infected for the rest of their lives, and they pass on the Borrelia to succeeding generations via the ova. So the relapsing fever clinical manifestations, it doesn't matter whether it is from a tick or from louse. Clinically, the illness is the same, and the main feature is fever, and it is fever which comes and goes, that is recurrent. So it's characterized by febrile episodes and a febrile period. So febrile period, that is when the fever, the Borrelia are present in the blood circulatory system of that person, and because of their presence, the immune system is stimulated. So the immunological defense mechanisms are stimulated, and the antibodies, the antigens on them are detected, and antibodies are formed to eliminate the organisms in the circulation at that time. So the antibodies, once they are formed, they are released, and together with other immunological defense mechanisms, they act to eradicate the spirochetes in the blood, and they do so As a result, there is marked reduction in number. So they managed to reduce quite a big percentage that would cause a marked reduction that would have some significance. So fever subsides because of the small number that might still be there. And then after 4 to 10 days, the Borrelia reappear in the blood in large numbers, causing a relapse. So that reduction is not complete. They don't capture all the Borrelia. There are few that are left, and we are going to know why they are left. And as the fever subsides, the number is still small, but they continue to multiply. So that at least 4 to 10 days, the number has reached a proportion that is enough to cause fever. And it is also significant in the blood. So the Borrelia reappear in the blood in large numbers. So the process it happens repeatedly. The febrile episode and the febrile periods occur repeatedly. It's possible eventually the body healing system catches up and eliminates them. And for most people, it's just fever, and a febrile and a febrile episode. For a few, it says that they can affect some of the internal organs and cause damage to those organs, but for the majority, it's just fever and that comes and goes. So at the time there's fever, there's a large number of the organisms in the blood circulatory system, and from that large number, whenever one has bacteria in large numbers anywhere, some of them undergo mutation. And for this particular case, in the a few Borrelia undergo mutation, it would cause, which would affect or cause a change of the antigenic structures. So that mutation targets the antigenic components, the parts that are recognized by antibody-producing cells. So these are affected, and when they multiply, now the ones that are being produced have a different antigenic structures from the original one. So from that mutation, they develop new antigenic determinants different from the previous one, and these new organisms with new antigenic components constitute new strains. So this phenomenon of changing antigens is known as antigenic radiation. So these organisms, the released antibodies, initially when you have a large number, the antigenic component is recognized and specific antibodies formed against them. So when they are released, they look for the antigens that stimulated their formation. So released antibodies recognize the antigenic structures of the larger number of the infecting strains at the time, and are effective in their elimination, but the few Borrelia that have undergone mutation are not recognized. So they are the ones that remain. Initially few, they multiply and cause relapse. So that would be the explanation for the fever that comes and goes with regard to Borrelia. So for lab investigation, one needs to demonstrate Borrelia in the blood. And the best time to demonstrate that would be when there is fever. That is when the organisms are plenty in the blood circulatory system, or one stands a higher chance of detecting them. So if we say plenty, at times it's not so easy to capture them, but the chances are higher at the time when the fever is on. So the methods used, one can take blood and examine it directly, a drop of blood under the microscope. Hopefully, one who wants to see the organisms, a spiral motile organism with that blood. Secondly, dark field of this contrast microscope can be used. and explain the dark field, but the phase contrast also operates under the same uh, way. In the same way, it has its unique um, condenser that limits the light and focuses more on the light from the organism that enables one to see that organism. So that field of phase contrast microscopic examination of the blood, and this is a more reliable method. And then thirdly, one can make a peripheral blood film, uh, dry and stain. Here, say one can use Bianca or right film, and one will see the Borrelia in that specimen. For the antibiotic susceptibility, it's also it, penicillin is also effective against the borreliosis. Then the third genus is Leptospira, and it has that morphology of a spiral organism with closely set coils. Again, we say this is subjective, the people who examine all of them at the same time, but it is also said to have a characteristic vigorous motility. It is visible if one uses dark field microscope or phase contrast microscope or electron microscope. Again, it is said to be the most narrow, the narrowest or the smallest. But it is also stainable by special methods, one of them being the fluorescent staining methods. Its growth in the laboratory, small as it is, it can grow in the laboratory. And culture is one of the methods of investigating for infections where Leptospira species is suspected. So, organism associated with human disease can be grown in specifically formulated artificial media. These are available if one is looking for them. They could be in a liquid form or semi-solid media preparations, and they are grown. These media are inoculated and incubated, and as in air. So the organisms grow as obligate aerobes. And their growth is slow, especially when they're being grown for the first time from a specimen, that is a primary culture. It can take 6 days up to 14 days, and sometimes even longer than 14 days. So the species of Leptospira, there again, it's a genus that has a mixture of organisms. There are some, basically they are saprophyte, they have that saprophytic existence, but they are also a mixture. There are also some of them which might be saprophytic at some stage, but are potential pathogens. So when it comes to species, there are only two species in the whole genus, and they are morphologically similar. One cannot distinguish each species from the other. And each species is separated again into serotypes. Many serotypes per species, and those serotypes, they have names. So you'll have a species name and a serotype name. But if we look at the two species, there are two. One is called Leptospira interrogans. And it is in this species, one of its serotypes is significant as a potential pathogen for humans. With there are many species, and Leptospira interrogans, and it's many serotypes in the species Leptospira interrogans. And among the many serotypes, there's one which is a potential pathogen for humans. Then the second species is Leptospira biflexa. And this particular species the serotypes are adapted to the saprophytic life. Most of them are all of them are mainly saprophytes, and they're found in various parts of the environment, including water. But he said they are morphologically indistinguishable. So if one detects an organism, one needs to know which species it is. And what, if it is found to be Leptospira interrogans, one wants to know the serotype. Because there's one serotype that is a potential human pathogen. So the transmission and clinical significance of Leptospira interrogans serotype that is associated with human infection. This particular one is parasitic to small animals, but it's also capable of infecting the bigger animals. Other animals, including domestic animals, should they be exposed. So infected small animals include the rodents, and they constitute the reservoir. They just harbor the organisms in their kidneys and occasionally pass it out in their urine. Otherwise, they are healthy. It doesn't cause any disease in them. And they pass it out, and from there can cause contamination of water or damp soil. So water or damp soil where are infested by rodents. Some of those rodents might be carrying this particular organism and might be passing it out and contaminating the water and the damp soil. So accidental transmission can occur to humans should one get into contact with contaminated areas or areas which have been contaminated by the urine of those animals that are passing out the organisms, or if one gets into contact with the urine itself, the animal carrier. This can be direct contact, which is unlikely, or it can be more likely to be through contaminated parts like soil or water. So once a person is exposed, the organism enters through broken skin like the others, or mucous membranes. Human infection is initially associated with invasion of the blood, resulting in septicemia. So it gets in and gets straight into the blood circulatory system. And consequently, the infection spreads to various internal organs, including the kidney, and of that particular individual. So the person might excrete the organism in the urine. So there are some people, because of their occupations, there's a higher chance of being exposed. Those people whose occupation involve working in damp places like plantations or mining areas or sewer places, which are infested by rodents. As we said, it's not all rodents. Some of them might be, and the place can be infested by rodents which are not carriers of this organism. But some might err in some areas, there might be some. So that would increase the risks of being exposed by those who are exposed to that environment. So the manifestations of that infection, the illness is referred to as human leptospirosis, and it is an illness of variable manifestations. Some people get exposed and they remain asymptomatic. Some people develop mild illnesses, some moderate, and some very severe illness. And where this is severe illness, because the organism is within the body, it can infect a specific organ and cause the damage to that organ. So the manifestations would involve the functions of that particular organ that will be deranged. So for investigation of human leptospirosis, one needs to detect the spirochetes in specimen from a patient. That is one approach. Another approach is serological tests, that is testing for antibodies. So one can apply both or apply one or the other. Among the specimens to be examined, blood is one, urine is another, and also tissue. The methods, again, one can do dark field microscopy for the spirochetes. This can be in urine or it can be in blood, and culture can also be done. We mentioned the media, that there are some formulated media that can support the growth of leptospira interrogans, and one can purchase that if needed and incubate appropriately and for long enough and examine the cultures. So it could be a liquid medium or a semi-solid medium. And then serology, this would be a test for an antibody to the specific serotype. And then the molecular methods, DNA detection, and also amplification tests. And then for antibiotic susceptibility, it's like the other spirochetes, the antibiotics remain the same. And since it's something that might be related to or suffer this one's occupation, there are measures that can be applied to prevent the infection. And they could target the animals that could be potential sources of the infection. They could also target the person if they can wear some protective gear, clothing or boots, or be made aware that there's such a thing and they need to protect their skin from getting into contact with that soil or water that is in the area where they work. So that is the Spirochaetes lecture, and I wish to stop there. Sure. Sure. No chats. So it's over, I think, for today. Unless you have something that you wish to say.